Hi, everyone. Welcome back to the Live Lounge. My name is Rachel, and I am excited to be hosting you again today for the product demo. So yesterday's topic, we focused on going big and using the PromethION devices and sequencing the larger human genomes. And today we're going to take it all the way to the other end of the spectrum. And today we're going to talk about using Oxford Nanopore technology for small genomes that affect all of us, bacterial and viral genomes. So by the end of this demo, I hope you will all understand how our technology is a perfect fit for these smaller genomes and applications like understanding what microbes are present in an unknown sample and characterizing pathogens and antibiotic resistance in known samples. And just like yesterday, at the end, we'll be joined by a guest expert who is going to talk about her research and how she applies the technology in really neat and interesting ways. So, as far as most of us know, bacterial and viral genomes are ever-relevant topics in this day and age, and they're only becoming more relevant as antibiotic resistance grows and international travel brings us all into frequent contact and brings pathogens from one place to another with relative ease and frequency. So, as you can understand, having the technology and tools to quickly assess and monitor this has become critical. So today I want to discuss how the technology is vital for related research areas with these small genomes, such as infectious diseases. And infectious diseases are a big focus for us here at Oxford Nanopore. And we've taken a lot of the learning from our work with SARS-CoV-2, and we are now really interested in expanding this technology to other areas within the metagenomics and pathology space. Some benefits of the technology that we can all appreciate are the rapid turnaround time and sample prep, showing that you get a very quick time from sample to answer, portable device size, and long reads that can completely resolve microbial genomes. These tools are also proving vital for microbiology and infectious disease due to the lower cost per sample and the concentrated effort for surveillance and containment that is enabled by the global deployment of these devices throughout the world and their accessibility. And I think perhaps most excitingly for us here is that it helps us on our mission to democratize sequencing for anyone, anywhere, which is our company's mission and something that we are all quite passionate about. So today we're going to focus on the MinION flow cells and the devices that use them. I'm sure most of you have seen and or touched the new MinION Mark 1D and its customized iPad case. So this setup is both portable and powerful, and it can enable the analysis of both known and unknown samples. Coming soon, we are going to release an isolate sequencing protocol that is end-to-end. This protocol uses our rapid barcoding kit with the V14 chemistry, and this gives you the ability to multiplex small genomes on one MinION flow cell while still getting 30x or more coverage. The rapid prep accepts samples from many origins and input types, and this unlocks a lot of capabilities across the fields of epidemiology, infectious disease, and metagenomics, like I've mentioned. One new area that we're taking this technology is wastewater sequencing for pathogen surveillance. One reason why the technology is a great fit for this application is due to the rapid prep and turnaround time, which enables insight into emerging new variants, as well as large pathogen transmission events. Additionally, we can get geographical information about pathogens that is not possible with the other methods that are not sequencing based. This kind of information helps us to inform policy at scale, and it also helps us with the goals of current containment of any pathological events and help prevent and mitigate in the future. However, what if you wanted to get more into the unknown? Much of the microbial world is hidden to us, and while I wish we could just, pick up some dirt and know what's there, the reality is that we can't. So we need effective tools to uncover unknown microbial and viral pathogens. With the data gathered from nanopore sequencing, you have excellent input to bioinformatics tools to help solve these challenges of the unknown. We have epidemy solutions, and they were just announced yesterday. We have now an automated de novo assembly of bacterial genomes, and an in-depth metagenomic analysis as well. So the bacterial genomes workflow, as you can see on the screen, provides assembly, variant calling, and annotation in a really nice report and easy-to-use format. This workflow is a great companion to the isolate sequencing protocol that I just mentioned, and it also is able to resolve complete genomes and exogenous plasmids while creating high-quality reference assemblies, de novo assemblies, and this also works with multiplexing. The metagenomics workflow works with inputs from amplicon-targeted or shotgun metagenomic sequencing. And this helps us detect unknown and novel pathogens that often can escape traditional methods of detection. So if you want to learn more, please do not hesitate to go downstairs to the data analysis DAT cave and talk to our talented bioinformaticians, because they would love to tell you more about these and all of our workflows. Now, the more exciting part of the talk, I would like to introduce Natalie Ring. Natalie is a postdoctoral fellow at the Roslin Institute in Edinburgh, and she is a senior clinical research scientist at the Royal Dick School of Veterinary Studies. Natalie's research focuses on developing methods for culture-free prediction of antibiotic resistance and veterinary infections. So, without further ado, please welcome Natalie. Welcome.
Hi.
Hello. So, would you mind giving a brief overview for those who have not yet seen your Secret Cinema presentation?
Yep, so my work. So actually, I started using Nanopore to sequence Bordetella pertussis back in 2016, which was my PhD project. Bordetella pertussis causes whooping cough. And the reason we were using Nanopore back then was because it's got this really repetitive genome, and we needed long reads to resolve the genome. So we were using 12 barcode kits back then, so we were doing 12 isolates per run, and we were comparing these genomes, and lots of interesting stuff popped out, like rearrangements, duplications, that kind of thing. Since then, I've moved up to Edinburgh for my postdoc, and we are developing ways to metagenomically extract DNA from veterinary samples and then really, really quickly sequence them. And what we're looking for in the DNA is to identify what species is causing the infection, and then assemble genomes from this and work out if there's going to be any AMR in the infection.
Awesome, super interesting, and I love your little diagram. So, given all of that, how has Oxford Nanopore sequencing enabled you to predict antimicrobial resistance over traditional methods?
So the traditional way that infections are being diagnosed in the veterinary world is very similar to in human clinical practice. So it's culture-based. They will get a sample from a dog, say, and they'll grow it overnight, see what species they think it is, and then they'll grow it on a bunch of different antibiotics and see what grows and what doesn't. The way that we're doing this is literally everything comes from that DNA, so we can get to the answer in 6 to 8 hours versus growing these bacteria several times, which can take maybe a week.
Cool. Do you foresee bringing this technology out of the lab and closer to these infections, or do you foresee it always being kind of a lab practice?
Actually, the next thing I'm focusing on, and the reason I have that second part of my job title, which is the senior clinical researcher, I'm going to be moving into the vet hospital, and we actually have a cupboard that is currently in the building that I'm turning into the Nanopore lab, where we will literally on site be doing the sequencing. And as the vets come across animals they think might have an infection, they will literally just send it upstairs to me, and we'll be running it all the time.
That's awesome. So being, the expert that you are, what is the biggest lesson you've learned, or what would you not recommend to people getting started with the technology for this or other applications?
For me, it's push it as far as you can, really. So when I first started, I was using one flow cell, one run, and then throwing the flow cell away. What I've learned more recently is that you can really, you can push the flow cells to their limits. I've done a lot of experiments where we'll do a small run, wash the flow cell, store it for a bit, wash it again. We can reuse flow cells like 10 or 11 times, and it's a really cost-effective way of using them again for different samples.
So 10 or 11, is that your record of reuse?
Yeah, but only because we're using a 12 barcode kit. So if we had more barcodes, maybe we could push it further.
That's awesome. This might be a silly question, but what's the lowest core number you've started with for great results on a reuse?
So we're only really looking from these samples for 100 to 200 megabases, so you don't need a lot of cores. So after maybe 10 washes, we're starting with maybe 300 pores. And you're still getting good data, it's just that it takes a little bit longer to get it. So when we're going for fast results, that's when you have to really say, okay, we need a fresh flow cell now.
That's awesome. And then I guess where are you going next with this tech? You did mention going on site. Do you have plans past that?
Yeah, so what we've developed so far was focused mainly on dogs. It was a Dogs Trust funded project. We've optimized the way that we extract the DNA for skin swabs and for urine samples. But I've got this ultimate goal where, with a very similar protocol, you could basically do this process from any kind of clinical sample, any veterinary sample. So blood samples, tissue samples, basically anything comes along, not just dogs, but any animals. There's one vet in the hospital that works with bears, and she's quite keen to do this for bear TB, which is very cool. It's one of the really cool things about being in the hospital is the vets will have all these really cool ideas, and we're just gonna try things.
That's great. I bet it's really great to work with all these animals. So thank you so much for that info. And I'd like to open the floor for questions of both me and Natalie, if anyone in the audience has any.
So thank you. We are in the same boat. Do you like better gram negatives or gram positives?
Oh, half and half. So gram negatives are quite easy to get out of the samples, but actually when it comes to identifying the species, they're all quite similar to each other. Whereas a nice gram positive, you can stick that in Kraken, say, and it's very clear what species that is. So they each have their positives and negatives.
No other questions? Oh, right there. Perfect. 1 second.
Do you have any tips or tricks for extraction and how they influence your representation of genomes, especially gram positives?
So I kind of went for a slightly different extraction to a lot of metagenomic extractions, which are bead bashing based. And I use a cocktail of lysis enzymes instead, which is much gentler in the DNA. You get longer DNA at the end of it. And actually, I've done quite extensive testing on gram positives and gram negatives, and it does seem to extract equally well. So my tip would be, yeah, you don't have to use the more vigorous physical methods of extracting the DNA.
Well, I have a question that just came to me. In terms of read length, are you here looking for the longest? Do you shear? What works best for your application?
Any length, really. So we tend to get an average of maybe 3,000 to 5,000. So they're not the longest reads, but we're not being particularly gentle with the DNA. What we really want from the DNA is to be able to identify species, which any length works, and then ideally see those AMR genes. And again, yeah, low thousands is fine for that.
Nice. One more from the audience? Well, I am sure Natalie will be happy if you come chase her down later. So just to summarize before we end, today we went over the application of our technology for small and often pathogenic genomes. We talked about the benefit of portable devices, which clearly Natalie will take advantage of, bringing them closer to the infections themselves. And we talked about the new and improved Kit 14 chemistry and the fast analysis with our epitome solutions. So thank you all.